So as we mentioned, when we're talking about long-distance signaling with hormones, there's usually three steps that occur. First is the reception, which we've talked about can be done by either membrane receptors or intracellular receptors. And then we talked about transduction and response, and that's what this video is going to focus on. So usually when we talk about transduction, the receptors for the signal molecule are plasma membrane proteins, and they receive the signal, and then that ligand binding signals a whole long series of steps known as a transduction cascade. And this is a multi-step pathway. The reason why this is important is because a few molecules can produce a very large response using these multiple steps. The signals can either be amplified, meaning that there's a greater response, or they can be distributed to other cells more readily. And so this results in a much higher level of coordination and regulation than a simpler step system. Some examples of how transduction pathways are used include opening and closing ion channels, alternating gene expression, such as with transcription factors, as well as the alteration of enzyme activities and controlling enzymes. So this cellular communication, these transduction cascades, are very important to all sorts of things that happen in biology. And all of this occurs through the regulation of proteins. So most of the time, the way that the response is received is by regulating a signal from a receptor to protein to protein to protein, almost like a domino effect. And finally, the protein that's responsible for producing the response gets activated. And typically, the way that this occurs is that at each step in the transduction cascade, the signal is transduced to a different form, which essentially is visualized by a change in the shape of the protein. So if you remember, we talk a lot about structure to function. You know that the structure of proteins greatly affects its function. So in converting its shape from an active or inactive form to the opposite results in a differing response. One of the most common methods of changing shape of a protein has to do with phosphorylation and dephosphorylation, which is one of the most common mechanisms, like I said, for regulating the protein shape, structure, and therefore their activity or their function. Phosphorylation, if you remember, just has to do with the addition of phosphates. So protein kinases are enzymes that help transfer phosphate groups from ATP to a protein, phosphorylation, and they often result in signaling a phosphorylation cascade, which is essentially a signaling pathway that changes the shape of proteins using phosphorylation and dephosphorylation, all the way going back and forth, changing proteins from an inactive form to an active form until you get to the protein that needs to produce a response. So one example of a way protein kinases are used is for regulating cell division. So here's kind of a whole picture example, and you can pause here if you need to, but we'll start at the beginning. We've got our signaling molecule comes in and binds to the receptor, and that ligand binding activates a protein here. The inactive protein kinase gets converted to an active form, and then that's going to result in the phosphorylation of another molecule, and we're going to end up getting another active protein kinase, which is going to keep going down, and then eventually we're going to phosphorylate this inactive protein. The protein kinase is going to phosphorylate that and then cause it to be active, and that's going to result in a cellular response. So you can see kind of the whole picture here once again, but basically the protein kinase phosphorylates other protein kinases until we eventually get to the protein that we want to produce a response. The opposite example would be a protein phosphatase, and these work similar to protein kinases except for they remove the phosphates from proteins, aka dephosphorylation. And oftentimes These protein phosphatases are used to turn off a signal transduction pathway. And another important role of these phosphatases is they help to make the protein kinases reusable and reversible. That way the cell can respond to the signal again if necessary. And so these kind of act as, the phosphatases and the kinases act as a molecular switch, and whether they're working or not depends on the balance between active kinases versus active phosphatases in the cell. And in using this switch, we were able to regulate a variety of cellular activities. It's almost countless how many different things in the body and the cells depend on these two processes. Another important note is that there are small molecules and ions that are also used as secondary messengers in cell communication. So the first messenger is always the signal molecule or the ligand that binds to the receptor. That's always what starts the cascade. But typically there's these second messengers, which are small, non-protein water-soluble molecules or ions that spread through the cell by diffusion. And the two most common ones are cyclic AMP and calcium Ca2+ ions. So one way we've seen this used is with the regulation of epinephrine. So when epinephrine binds to the plasma membrane, that results in an increased concentration of cyclic AMP or CAMP, which we talked about is a widely used second messenger. That results in the epinephrine binding to a G protein receptor, which causes the activation of something known as adenylyl cyclase, and this enzyme converts ATP to CAMP. And once we have that happening, we're going to get lots more CAMP, and CAMP is usually going to activate protein kinase A, which, remember, the kinases phosphorylate proteins. That causes a response, and you end up getting all the other proteins changing shape, and you'll see a cellular response from that. So here is a picture of the first messenger, such as the hormone epinephrine binding to the GPCR, the G protein receptor. That's going to activate our G protein, which is going to bind to the enzyme adenylyl cyclase. Once that happens, we get the conversion of ATP to CAMP, which acts as a second messenger, and that's going to activate protein kinase A, which then is going to phosphorylate many other proteins, and eventually, after many long cascades, give us a cellular response. So that is transduction. That's probably the most complicated step in the process. And then finally, the third step, we have the response. And this is really what happens as a result of the transduction pathway. And our transduction pathways are used to regulate all sorts of cellular activities. They can range in the function of what the cell is going to do, but ultimately these long pathways cause some type of response and make the cell do something. This can either happen in the cytoplasm of the cell or the nucleus. And some examples of what these responses could include might be the synthesis or making of enzymes or other proteins. A lot of times this is because those pathways signal to turn genes on or off in the nucleus. So if you think about our transcription factors, those are kind of the results of perhaps a long transduction pathway, and that transcription factor, helps regulate the expression of different genes. We also might regulate protein activity, which might have to do with like proteins outside of the nucleus, for example, might be telling them what to do. So either making proteins or regulating how they behave. So this is just one more example. If we have our growth factor signaling molecule binds to the receptor, so once again our growth factor is the ligand that binds the receptor, we end up with a phosphorylation cascade where all these proteins get phosphorylated. And in this case, through this transduction pathway, the end result is a transcription factor, which once it's been activated can bind to our promoter and then result in the transcription of a gene. That would be the response. So this kind of just zooms in a little bit more, just what we were talking about, that you can see right there. And other pathways that might be used to regulate the activity of enzymes. So once again, instead of them being made, just talking about their activity. And this would be, for example, the opening or closing of those ion channels like we talked about, or perhaps a change in cell metabolism. And that is all for the cellular communication section.